Yeah. Okay, so thank you for putting up with our technical difficulties for a little bit, and also for a little news interview that you got to deal with the media when you can deal with the media. So thank you all for braving the lovely weather this evening to come to the first talk in the Geology lecture series this year. And this is the next talk, so if you've got some place that you can post these, I'd be glad to send some off with you. If you would like your very own little list of some of the future events, you've got these little yellow sheets out there as well. Hey, I didn't drop them. And if you didn't sign in, if you could take a minute when you leave to go ahead and sign in, it actually helps us justify this as far as telling people how many people participate in this type of event. So it's my pleasure to welcome you all to the first talk. And before I introduce Dr. Davis, I'm going to just give you a quick rundown of some of the future events. We've got another Dr. Davis coming on the 30th of November. He's going to talk about the Ghost Coast. So fall is actually kind of a really classic Halloween event between Saber-Toothed Salmon. We're going to actually find out if it's real or not in a couple minutes. And Ghost Coasts, and if you tune in February, we're going to have a speaker coming down from Portland State, Scott Burns, talking about the Brett's floods through the Columbia. We're going to tune into mammoths at the end of April with Dan Fisher from the University of Michigan. And we've also got a speaker not on the list, May 18th, that's going to be here talking about major giant submarine landslides. So all that stuff is going on. One thing, free events are not always free, and I'd like to thank Oregon Resource Corporation for continuing to be a sponsor, as well as the college, and we're live streaming these talks now, as well as archiving them, so without the tech support in this hall, we wouldn't be able to do all that. It's my pleasure to introduce to you this evening, fresh from the East Coast, where he was attending the Geological Society of America meeting just a few brief hours ago, Dr. Edward Davis, who's coming to us from the UO Museum, and he's come to us also previous to that from UC Berkeley, one of the top paleontological schools in the country. And so without further ado, because you actually want to hear about saber-toothed salmons, not about me, is Dr. Edward Davis. Thank you.
Thank you very much. So not only did I bring back a lot of new ideas about paleontology from the Geological Society of America meeting in Charlotte, I also brought back a cold. So I'm going to try to make it through tonight without coughing too much, but please forgive me for this. Okay, so talking about Oregon saber-toothed salmon. I mean, how many people heard that and thought that I must be making it up? Yeah, a few people. Oh, you guys trust me. That's good. No, it is a real animal. It was a real animal, I should say. And so what I'm going to do tonight is try to use this ancient, extinct animal to connect you guys through your own experience to Oregon's deep time, and to think a little bit about how we can use the evolution of organisms like this to help us understand how we can anticipate ecosystems changing in the face of changing climates. Okay? I want to make sure that I give my proper respect to my colleague Brian Sidlauskas from the Oregon State University Department of Fisheries and Wildlife. Brian is a fish biologist, and my work with the giant salmon wouldn't be possible without his expertise. So I want to make sure that we communicate his role in the whole action, and also make it clear that we're collaborating between these two rival universities, something that we hope to do a lot of in the future. Okay. Smilodonichthys rastrosus, what is it? It's this giant fish, okay? Projected up on the screen, it looks even bigger than it actually is. But you can see from the scale bar and this figure here from the original 1972 paper that the thing's pretty big, okay? So this is just the skull. To give you a reference, this is where the eye would be, okay? And here's the mouth, and then this little dotted line here corresponds to this other fossil that's illustrating the saber tooth, okay? So one of the tragedies here is that the fossil that we have the complete skull for doesn't actually have the saber tooth on it. So we know that that's there because of other specimens that preserve it. So that's 5 centimeters. The skull itself is probably about this big. So we reconstruct the animal as being a little bit over 2 meters long, so it's probably between 6 and 10 feet long, depending on the individual animal. So if you can imagine a salmon that was like 10 feet long and probably weighed 300 or 400 pounds, maybe more, that's what we're talking about here, okay? And we find it in the fossil record from the latest part of the Miocene and the earliest part of the Pliocene, so from right about 5 million years ago, when the climate of the world was very different, but it was living along the Oregon coast, living along the Pacific coast, and actually doing some things that are very similar to what salmon do today. In science, we have to ask questions because science is about answering questions with new data. And so I wanted to have a series of questions to sort of start the talk out and get you guys thinking. And as we go through, we're going to try to answer all these questions today. So first of all, why did Mr. Fish get to be so big? We call this particular specimen with the skull Mr. Fish. And I'll explain more about why we call him Mr. Fish in a little bit. Then I want to know, why did it have saber teeth? Okay, so it could have been really big without having saber teeth, and it could have had saber teeth without being really big. So those are two separate but possibly connected questions. How different was the environment at that time? And then how could it, in this case the saber-toothed salmon, or other fossils, inform our ideas about making policy in the face of changing climate? Okay. Excuse me. So, how many people here are familiar with Ray Troll's art of the saber-toothed salmon? If you're not, you should get to be. Ray Troll is an excellent paleo artist, and I'm using all of his images tonight with his permission, but you should take it upon yourself to visit his website and see some of the other work he's done. But Ray Troll is a Pacific Northwestern paleo artist, and he has a number of really neat illustrations that kind of show how impactful the saber-toothed salmon can be in sort of popular cultural context too. So this is one of his reconstructions of this animal, sort of in context with a saber-toothed cat for scale, just to give you an idea of how big it might have been. Here's another one. I like this one. So obviously there weren't any anglers 5 million years ago to get pulled away by a saber-toothed salmon. But this is a little idea of what it would be like trying to capture one of these animals if you were able to get a time machine and go back and go fishing for them. So just, to give you an idea, bigger than your average person. This is a really huge animal, and so it's intriguing to try to understand what role it would have played in its ecosystem at the time. Yeah, oh, and then this reconstruction actually has it as much as 1,000 pounds, so we're talking about half a ton for this particular proportional reconstruction. Okay, so the one main specimen that's been studied is this skull that we call Mr. Fish. And Mr. Fish is a type specimen of this species. And so a lot of people don't know this, but in biology, going back to the time of Linnaeus, we've had this legal way that we deal with names, with scientific names. So whenever you get a scientific name, something like Oncorhynchus or Smilodonichthys, rastrosus, or Tyrannosaurus rex, okay, those scientific names are tied to individual specimens. So whenever a scientist discovers a new species, a species that hasn't been known to science before, he or she writes it up in a paper and publishes it, and when they do, they have to pick one specimen that's going to be the specimen that's tied to that name forever. Okay, so ideally you want to have a really complete specimen, but sometimes the best specimen you have is just an individual tooth. But it's a tooth that's different enough you know it's something different. In this case, Cavender and Miller actually were able to get this nice complete skull out. And so that skull and some of the associated parts of the skeleton became the type specimen for Smilodonichthys rastrosus, okay? If you look at something like Tyrannosaurus rex, that also has a relatively complete skeleton for its type specimen. Excuse me. So anyway, this particular specimen was collected back in 1964. Actually, some collecting went on as early as 1961 from this Gateway locality in Jefferson County, all right? And this uniquely Oregon fossil comes from the Madras Formation, something that's called the torrential beds in the publication. And the reason why is because, as you can see in this illustration here, the guy is digging in this rock wall. It's actually a great big conglomerate. So it's made of big bunches of pebbles and actually big cobbles and stones all thrown together. Torrential beds, that name comes from the idea that these were all brought down in one torrential giant mudslide that covered everything. And so when you go to the site, you actually find the salmon coming out right at the base of this big slide. So it's like the animals were actually in a big pool or a very slow-moving stream, and they were spawning, and then they were all covered at one time by this landslide, okay? That's the debris flow I'm saying here. And this particular site is about 8 million years old, so that would put it right at the end of the Miocene epoch, okay? Now, the University of Oregon crews started working there back in 1961, and there was a big hiatus, but last year we actually went back to this site again, and new fish have started to come out. So we've actually started to recollect in this place. Here's a couple of our students from the joint UO-OSU dig that we had last summer. The landowners were very courteous to let us in there, and they also allowed us to have a salmon smoke at the site. So while we were collecting salmon, we also smoked some modern salmon and had a great party there. Okay, you guys are a tough audience. That's a little bit better. Okay, so this type specimen was collected in the early 60s by University of Oregon crews. It was in our museum in Oregon. But we don't have any fish specialists here, okay? So needed somebody who really had expertise in fish. So in 1966, this man, J. Arnold Shotwell, who was the director of the UO Museum at the time, packed it up and sent it to Miller at the University of Michigan, okay? And Miller was an expert in fossil fish, and he proceeded to publish it. So I got the University of Michigan seal up here. Went to Michigan in 1966. They published it in 1972. Excuse me, so 6 years later it got published. And it was actually wonderful. They published it in the University of Oregon Bulletin. So we have copies of this available through the University of Oregon Museum if anybody's interested. It's a great paper, and it's got some really wonderful line drawing illustrations of the specimen. But it was limited by the abilities of the technology at the time, and so I'm hoping that we'll actually be able to improve on it, as you'll see. That was not the end of it. It was in Michigan. It got published in the University of Oregon Bulletin, but stayed in Michigan, and actually stayed in Michigan until 1986. So one of the things that happens with fossils when they go on loan to other museums is that they can sometimes get forgotten, all right? So you need the fossil so that you can do your paper on it, and you're doing other things. You don't want to send it back yet because you might need to do some more stuff. It gets buried in the back of the drawer in your cabinet, and then you retire, and then somebody else gets that office, and the fossil's still there, and it can actually stay for a long time. So you got to see, for 20 years, Mr. Fish lived at the University of Michigan. And in fact, I have paperwork that indicates they were about at the point where they were going to put a University of Michigan museum number on the specimen. That would not have been good for us. So fortunately, our own Bill Orr, who is now basically emeritus director of the fossil collection, took it upon himself to get the specimen back. So it's been repatriated. It's at our museum now. Every now and then we bring it out for exhibits, but most of the time it lives secure in a cabinet where it can be safe from degradation. Okay. And that's basically where it stood until 2010 when Brian and I got together with some people at Oregon Imaging Centers in Eugene, and we actually pulled it back out and we took it to get CT scanned. And this is sort of the latest episode in the history of Mr. Fish, and that's where he got his name. Okay, so we took him to Oregon Imaging Center at the suggestion of Lee Michaels, who is a museum volunteer at the University of Oregon Museum and retired director of Oregon Imaging Centers. And we got a couple other people, Derek and Nathan here, to help us with the scanning. They're used to scanning people, so it was very exciting for them to get to scan this fossil. The biggest thing that they were excited about was that when they work on people, they have to keep the energy pretty low in the CT machine because they don't want to fry anybody. But it turns out that you can turn the safeties off on the machine and you can jack the power up really, really high. Excuse me. And so that was actually quite the fun thing for them. We had to click through a bunch of warnings. And kept saying, are you sure that you want to do this? But we had to name it, okay? So the computer won't do a scan unless it has a patient's name registered in it. We had to come up with a name, so we called it Salmon Fish. And so all of the records that came out of it were like Mr. Fish, Mr. Fish. And so we just started calling it that because that's what the computer thought of it. All right? So to give you an idea, anybody here ever had a CT scan? I'm sure people have done this. I know I have. I got screwed up my knee at one point, had to get scanned. It's not any fun. You had to sit still while the machine goes and makes a bunch of noise. Okay, but then you get these cool 3D pictures of the inside of your body, which is awesome. Okay, the salmon didn't care about sitting still. It's already dead, so it doesn't mind. It was able to fit, I can't actually, I'm going to have to remember this and sort of point around the corner here. But as you can see, we were able to put it together. They had some CT inert foam that they use for other scanning activities. It doesn't show up on the scans, and so we were able to prop it up together in the proper position and then actually run it through the machine. And because it doesn't have to hold still, as a person, they were able to actually slice it a lot finer than they do with people. So it could take longer, and they turn, like I said, they turn the energy way, way up. So when you usually do a CT scan, everybody else goes out of the room because there's x-rays, you don't want to get x-rayed. Well, we had to do that here too. But when we got done and we went back in the room to get the specimen out of the machine, it was like 15 degrees hotter in the room. Okay, so it was emitting enough energy that the coils on the machine got hot enough that it overwhelmed the cooling system for the room, actually made it perceptibly warmer. But you know what? Mr. Fish don't care. So it worked out. Actually, because they were able to use the high energy, we were able to get a lot of really good detail on the inside of the specimen, and that's the whole goal here. So this specimen is really delicate. It's basically the original bone material preserved for 8 million years. So it's very fragile. It hasn't been, a lot of fossils are what we call permineralized, where the bones have been impregnated by other minerals, and that actually makes them stronger in the end than just bone. That hasn't happened here. So in the end, it's actually a lot like dry bone. You might have ever had fish bone that sat around for a long time until it gets really dried out. You know, they can be flaky and kind of, yeah. So that's what this specimen's like. As a consequence, we can't dig down into it any further to find out what the internal anatomy is like. And one of the things that's really important for understanding in paleontology is how the brain is put together and how the different nerves and blood vessels go into and out of the brain case. We can actually understand that now because we were able to do the CT scan, and we can take it apart inside the computer while keeping the actual specimen intact for future researchers to look at. One of the other things I'm looking forward to is it's too fragile to reproduce this specimen. So a lot of the ways that we communicate unique specimens to other museums is by making casts of them. Okay, so we'll take the specimen, we'll make a silicone mold that's a lot like the one that a dentist makes of your teeth, an orthodontist before they design your orthodontia. Okay, and a lot of specimens are robust enough for that process, but this specimen is too delicate. We could never make an actual cast of it this way. But by having the 3D information from the CT scan, by having these wonderful images, excuse me. Wow. Look at the fish while I cough a little bit here. So by having these wonderful 3D images in the computer, we can actually print out reproductions of it. So there are machines called 3D printers that will actually let you take a 3D computer model like this and produce an actual physical 3D version of it. And so what we're planning on doing is actually printing several copies of this specimen and sending them to other museums so that researchers at those museums will have access to them. And that way, people don't always have to come to our museum if they want to study this specimen. Okay, the other thing we can do is we can actually make the CT scans available to those researchers. So we can actually take the scan data and just send it over email. And that way the researchers can look at these things without having to come and actually visit the real specimen. And that makes it a lot easier for people to get access to it. Otherwise, it's difficult for people. They have to come all the way to Eugene, which is not bad, by the way. We want them to come to Eugene, visit the museum, talk, and find new specimens. But at the same time, getting this specimen out with wider exposure can only help the science. Okay? So, at this point, we have these wonderful CT scans, and we're actually putting together a team that's going to go through slice by slice, millimeter by millimeter, and describe the internal architecture of the skull and write that up as a scientific paper so that we have that available for other people in the community, and we can compare it to other modern salmon and see if there are other things hidden inside that are different that might tell us more about the animal's evolutionary and ecological role. Okay. So we know a little bit, actually quite a lot about its evolutionary history already, just from looking at the exterior morphology of the skull. Morphology is just a fancy science word for shape. So by looking at the way the skull bones are put together, we can actually compare it to some of the other salmon from today, and we can come up with an evolutionary tree, okay? So this Stearley-Smith paper is actually covering all different kinds of salmon. And I've just cut off one part of the evolutionary tree from that paper to show you where our friend Smilodonichthys falls in it. All right. Now, when Cavender and Miller published this guy back in 1972, they gave it this name, Smilodonichthys rastrosus, okay? And the critical thing for you guys is the Smilodonichthys. That's what everybody calls it. That's a memorable name. Smilodon is, of course, the scientific name, the genus name for the saber-toothed cat, the one that's found at Rancho La Brea and other places that's much younger. And then ichthys is a Latin word for fish. Okay, so basically Smilodonichthys means saber-toothed fish, which is what we usually are calling it. Well, it turns out that if you look at the evolutionary tree of Smilodonichthys and try to understand where it comes out, it actually comes out right here between the pink salmon, Oncorhynchus gorbutia, and the sockeye salmon, which is, I'm going to actually forget this. Oh, yeah. I'm not a fish biologist, so I apologize. Oncorhynchus nerca, okay? But the critical thing is that as biologists, we want all of our scientific names to reflect evolutionary history, okay? Evolution links all different aspects of the tree of life. And if we want our names to be meaningful, then those names have to reflect that evolutionary history. Okay? So what Stearley-Smith did when they realized that Oncorhynchus bracketed here Smilodonichthys was they sank the name Smilodonichthys, okay? So when you sink a name, that means that you say that that name is no longer valid. In this case, Smilodonichthys becomes a junior synonym of Oncorhynchus. A junior synonym means, junior of course meaning younger, and synonym meaning same name. So what they're saying is that this name right here isn't something that's evolutionarily distinct from this name that was older and so has precedence. So as a consequence, after Stearley-Smith's work, Oncorhynchus now becomes the genus name for the saber-toothed salmon. And Smilodonichthys gets demoted from genus level to what we call subgenus, okay? So when you look at a scientific name, usually you're only getting like Oncorhynchus gorbutia. You're getting the genus and the species. But there is a level between those two. We don't always talk about it, but it's the subgenus. When we have a subgenus, it's put in parentheses right after the genus like this. So we were able to save the name, but not as a genus, it's only a subgenus. And sometimes, because often this is what happens to genera when they get sunk, sometimes we call these the parentheses of shame. Okay, so Smilodonichthys has been relegated to the parentheses of shame. And when we do scientific work on this now, we have to be very careful to call it Oncorhynchus rastrosus. I have a hard time doing that because I think of it as the saber-toothed salmon. And Smilodonichthys has this wonderful ring to it. It's actually got some kind of name recognition, especially in Oregon. So, excuse me, you'll forgive me if I continue to call it Smilodonichthys in a kind of an informal context. But when we actually do scientific research, we end up having to call it Oncorhynchus, parentheses, Smilodonichthys rastrosus. Okay? But one of the things that that does is it makes it clear that this giant saber-toothed salmon is actually part of the same evolutionary group, the same evolutionary radiation as these two living salmon today. And that links it more clearly to those groups. So when we're thinking about this animal, we have to think about it in the same evolutionary context as these other guys, and we have to think about its role in the ecology as a modified version of the roles that these guys share. Okay? So that puts it in better context. Okay. So when we look at Oncorhynchus, Smilodonichthys rastrosus, where all do we find it? Well, it's always found on the west coast of the U.S. Okay, it's not found anywhere else, so it's definitely a Pacific Coast salmon, which fits with its evolutionary relationship to the pink and sockeye. And it's only been found at these 6 localities so far. So the northernmost locality is actually that one, the gateway locality where the Mr. Fish specimen comes from. It's also found in Warden, which is also in Oregon, and then it's found down in Drake's Bay in California, and also Point Pinole, and then a couple other California sites, Turlock Lake, which is an interior site, and then down near San Diego at Loretta Street. So what we're seeing is West Coast distribution, we're seeing a lot of these as inland. They're in some kind of a shallow stream or a shallow, quiet lake. But a couple of them, the Drake's Bay site and the Loretta Street site, are both marine sites. So the sediments there are clearly coming from ocean sediments. So that tells us that just based on where we find the fossils, this animal was behaving like the modern sockeye salmon. It was living in the ocean for part of its life, and it was coming in and it was living inland for part of its life. One of the cool things that's been done, actually by my colleague Paul Koch, is that he studied the isotopes in the vertebrae. So it turns out you can look at what different kinds of heavy oxygen and light oxygen are in the bones of an animal, and it can tell you something about where it lived and what kind of water it was drinking when it was alive, or in the case of fish, what kind of water it was breathing. And so by looking at the oxygen in the vertebrae of this animal, he's been able to determine that it really was being born inland in a stream, swimming out to the ocean, living in the ocean for most of its life, and then coming back up the streams to spawn before it died. That's really cool. It's really cool scientifically, but it's also really cool kind of aesthetically. I mean, if you can think about this 1,000-pound, 10-foot salmon swimming up the streams, bounding up the waterfalls, that's quite an impressive thought. And if you go back 8 million years ago, there were more than just bears roaming the countryside. There were these things called amphicyonids, which are colloquially called bear dogs. And they were proportionately bigger than bears, the same way that this guy is proportionally bigger than modern salmon. So you could imagine a situation where a 12 or 15-foot tall bear dog would actually be swatting a 1,000-pound salmon out of the river. Isn't it? That's a cool idea. That's a cool idea. Although, that's one of those things like, what color is it? We'll probably never actually be able to see. But there's a chance someday that we'll be able to do some studies in the isotopes in the bears and show that the bears were actually eating these giant salmon. That would be cool. And that's science. Okay. So, it's more than 6.5 feet long. It has these giant saber teeth. I put giant in quotation marks because they're actually not that impressive. They're impressive in that most salmon don't have saber teeth at all, but relative to something like an actual Smilodon, they're not impressive. So skull's about this big, but the saber tooth is only about 2 centimeters long. Okay, so they're giant in quotation marks, but it has saber teeth. That's still something. Why is that? How could that be? Okay, and in the context of evolutionary biology, we have 2 major processes that could be governing one or other or both of these particular aspects of the animal. We've got natural selection, and we also have sexual selection. And so what I'm going to do here is walk you through some of the ideas about these different kinds of selection so we can see which are the likely causes of these particular amazing features of this amazing animal. All right? Excuse me. So, I'm sure everybody here is familiar with the idea of natural selection. I'm going to walk through it really quickly because I think it bears on understanding why the animal's so big. Okay, so Darwin's initial idea about natural selection still pretty much stands up today, and he had 3 simple ideas that everything is built upon, okay, and 1 inference. So the first one is that individuals vary within a species, okay? So here's some really extreme examples that are a consequence of artificial selection by human beings, but they really illustrate this idea that individuals within a species vary. All right, so we've got the diversity of different kinds of dogs. All different breeds of dogs all still fall within one species, and all that we've done as human beings is pull out that natural variation and make it really extreme and obvious. And this is an example from plants with peppers. Okay, anybody can look around the room here and actually see tremendous variation in our human species as well. And anybody that's ever worked with animals or plants in the outdoors is familiar with the variation that you can see in the natural world. So that's not a big problem. Animals vary. Well, the next idea is that babies actually look a lot like their parents. Okay. When Darwin had this idea, he didn't understand why. He just knew it was there. Today we have a really good understanding of the process of genetic inheritance and actually all the biochemistry that goes into it. These are my kids. They look a little bit like me, I think. So And then the cat's illustrating the same idea. So within certain parameters, babies actually look a lot like their parents. They have the same characteristics as their parents. They're inheriting their characteristics from their parents. Okay? And then the last idea that's really important for natural selection is that there are always too many babies. All right? So, there's always the runt of the litter with the pigs. There's always too many babies. When we look at mice, this is extremely the case. Most mouse babies don't grow up, and the mice, kind of expect that. That's why they're having so many babies. And then, like with trees, if an oak tree produces 1,000 acorns in a year, and one of them actually grows up to be another oak tree, it's pretty good. All right? So there are way too many babies, and so what's happening then? Excuse me. Is that all the babies are competing for resources, and the ones that are able to use the resources most efficiently are the ones that grow up and get to make more extra babies. Okay? So those three ideas right there get you to the point of natural selection is happening. And then the other thing, the inference that Darwin had to make, and that we still make today, based on even more evidence than he had at the time, is that the Earth is very old. Okay, and today we know that the Earth is approximately 4.6 billion years old. Now, when Darwin was working, he had no idea how old the Earth was, and he had no expectation that we would ever be able to put a number of years on the age of the Earth. But he knew that it was very old from all the work that he'd done studying geology. And if the Earth is old enough and natural selection is working, then you can actually get all the diversity of life that you have just from natural selection. Okay? So in the end, natural selection is acting to optimize the characteristics of organisms generation by generation to their habitat. Here's an example of natural selection I really like. It's a little bit like artificial selection because it's a consequence of human action, but it's not on purpose. And some of you guys out there are going to be familiar with this if you've got lawns and you deal with dandelions. Okay, so dandelions, naturally, they usually have very long stalks on their flowers. Okay, so in a natural system that's undisturbed, dandelions are trying to fight for access to the air. They don't have to be pollinated because they're self-pollinating, but they need to get the little puffball up high enough that the seeds can be dispersed. It needs to get up higher than the other grasses around them. The highest dandelions that are above the grasses will have their seeds blown away and get the furthest, and that gives them an advantage. So getting to more new habitats, okay? Excuse me. So natural systems, I should say, systems that are not affected by human beings, tend to have a lot of really tall stalked dandelion flowers. Now, when you start to mow, what happens? All the dandelions with tall stalks get mowed off before they get a chance to reproduce. And if there's one dandelion in the whole lawn, one dandelion that has a short stalk that's shorter than your lawnmower blade, then it's going to be the only one that gets to make a puffball. And what's going to happen? All of a sudden, well, not all of a sudden, but over a very short period of time, all the dandelions in the lawn will be descended from that one dandelion, okay? And all of a sudden, you'll have a lawn full of dandelions that are reproducing even though you're mowing. So some people probably have actually experienced this in their own lawns. It's very infuriating, but this is just like a macroscopic version of what's happening with drug-resistant bacteria, okay? So you mow them all down, if any of them can stand it, then they keep going, and then natural selection kicks in, all right? This is a really cool example right here of natural selection. This is by our own Betty Roy from the University of Oregon, Department of Biology. There are a bunch of desert wildflowers that have these wonderful yellow flowers. But what's happened is that a fungus that naturally infects these flowers has actually been selected by the butterflies that pollinate the normal flowers. So the fungus now co-opts the genetic machinery of the plant and produces a false flower. This is wonderful. So the flowers, some of the flowers that you see on these plants are actual flowers that the plant is using to reproduce. And some of them are actually places where the fungus has stunted the growth of the leaves and produced a yellow color and actually produces the same scents as the flower, but not from the plant. The fungus actually produces the scent. This is part of what she's discovered. And so the butterfly is confused, and it'll actually try to pollinate these, and as a consequence, it gets the spores from that fungus on it and carries them to other plants while it's pollinating the flowers. So the fungus has been naturally selected by the actions of the butterflies, which were also naturally selected to home in on the flowers that were originally there, so the fungus is now producing a false flower that confuses the butterflies and actually parasitizes the plant. All right, I think that's really cool. That's the kind of complexity you can get over long periods of time with natural selection. So what about size? What about getting bigger and smaller in natural selection? Because after all, we are talking about Mr. Fish. We're not talking about dandelions. We're not talking about fake flowers from fungi. So what about size? Well, there's two pretty well-known phenomena that lead to dramatic size changes through natural selection. One of them is island dwarfism, and the other one is actually island gigantism. So, for example, here is an elephant from the island of Crete. This is not, of course, a modern elephant. This is an extinct species. But what we see is there's like, you remember that animal is behind it, but this elephant right here from the island of Crete would have been probably shorter than the podium, maybe even only about 1.5 feet, 2 feet tall, okay? So if you can imagine a little bitty miniature elephant. That's not the only place they found them. This is just the best picture I found of one, but there were some like that on Channel Islands off California. There's some like that off on Wrangel Island in the Arctic, some mammoths that got dwarfed. So elephant dwarfing on islands is actually a relatively common occurrence through the fossil record. Another cool example is the Channel Island fox in California. These foxes have been dwarfed by living on the islands. Okay, like I said, the other thing is island gigantism. So sometimes animals get bigger when they go on islands. So here is an example of the Flores giant rat. So here's a more typical normal-sized rat. And then on the island of Flores, which is famous for having the so-called hobbit fossil. I don't know if you've heard about that, but it's an early human relative that is dwarfed. Okay, well, also on that same island we have giant rats. As if rats weren't big enough. I guess that would be a rodent of unusual size. Anybody have that context? Okay, so why does this happen? Why do we get these dramatic size changes? And is that something that maybe is happening with the Smilodonichthys too, but in ocean context? Okay, so island dwarfism, what happens is when you're on an island with really limited resources, smaller sized animals can have bigger populations. And the larger your population, the more numbers of animals that you have in a population, the more stable it is. So there's actually strong natural selection in that case with limited resources for smaller individuals that can have more individuals in their group. So over time, your elephants shrink to pygmy size, and then you can have way more individual elephants, and that gives you an overall group stability genetically, okay? It's neat because you can show it with pretty simple algebra, and then it shows up over and over again in the fossil record. Okay, what about island gigantism? It's a similar kind of idea. When you're a small animal, thanks. When you're a small animal living on the mainland with predators, part of your defense against the predators is maintaining your small body size. But say you are able to migrate to an island where there are no predators, okay? All of a sudden, you're affected by that same optimization for number of animals for the island. And it may be that you're able to get fewer individuals and still have the same population. And bigger animals, excuse me, are often able to use resources better and to have better success in their reproduction. So bigger rodents often can have more individuals of their offspring survive per generation. And so what can happen is that you can go from small rodents that on the mainland produce lots and lots of babies, and most of them die, to on the island, fewer, larger animals that are actually more invested in their offspring and have more of the babies surviving. And so I think part of what's happening here is related to that same idea of changing size, changing resource use, and changing survivability of the offspring. So when we look at Smilodonichthys, we can actually see some really good analogies from modern animals that are filter feeders, really big filter feeders like the whale shark and the humpback whale, okay? And they're not quite life-size in this giant projection up here on the screen, but pretty close. And the idea here is that if you're a filter feeder and you're spending all your time filtering on these little bitty animals, okay, then you can be really, really big and you can get more resources in just one big gulp. Okay, and it's clear from looking at the way that the gills are structured in Mr. Fish's skull that he's actually really good at filter feeding. So most people don't realize this, but the sockeye salmon does a certain amount of filter feeding. Its two primary foods when it lives in the ocean are squid and plankton that it's getting through filter feeding. But Smilodonichthys actually has much more efficient filter feeding apparatus than the modern sockeye. So it's filter feeding, but there's another thing that goes into the reproduction, and this is why I went through that whole thing about island and gigantism. When Smilodonichthys is coming back to spawn, it's coming up the streams and then it's living in these little lakes and ponds, okay? So it's actually getting to this kind of inverted island situation where we were talking before about an island of land in the ocean. You can think about the spawning areas for these guys as little islands of water in the middle of the land. And it turns out studies of modern salmon indicate that body size is a strong predictor of the number of eggs that they'll lay and also the survivability of their offspring. So the idea is that these giant salmon are actually getting some selective force because they're filter feeding, and that being big is important for filter feeding, but there's also probably some selective force for them to be bigger bodied because bigger bodied animals can actually produce more offspring when they're spawning. All right, so I think that the two major factors that are going into the size of Smilodonichthys have to do with filter feeding and with trying to optimize their ability to reproduce in the lakes. And in my conversations with Brian Sidlauskas and other fish biologists, it's been suggested that some of the modern salmon probably would like to get to be really big too because it would help them produce better when they're spawning. But there's an upper limit on how big they can get to be because of the way they're using resources in the ocean. What's the critical difference between the ocean that Smilodonichthys was living in 5 to 8 million years ago and the ocean of today? Well, 5 million years ago, it was a lot warmer on Earth than it is today. 5 million years ago, we had a lot higher carbon dioxide level in the atmosphere than we did before we started producing carbon dioxide with fossil fuel burning. I won't say than we did today. We actually aren't quite to the level of carbon dioxide that we reconstruct for the end of the Miocene, but some of the models of carbon dioxide production put us about at that level by the year 2100. So one of the things that we're seeing is that we're actually headed back for that same kind of carbon dioxide situation at the least as we had when Mr. Fish was alive. Okay, and the suggestion I like to make, and this is a hypothesis, we're trying to find new ways to test it, but right now what we're suggesting is that the warmer oceans off of the Pacific coast at that time were producing more in the way of plankton, and this guy was actually using resources that weren't available at the time. As a consequence of that different resource base, it was able to reach the much larger size. So what does that imply? It implies that it's possible that in the future, with continued warming, we might actually reach a situation where modern salmon might have some selective advantage for a larger size, increased filter feeding, and we might actually see some modern salmon being pushed by natural selection back towards this kind of an ecology. All right, that's a series of hypotheses now. We're going to try to get more data and test them over time, but that's how science works. We start out with hypotheses. If they're good, we keep them. If they're bad, we kick them out. We come up with new ideas. Okay, but what about the teeth? What about the giant saber teeth? Well, let's talk a little bit about sexual selection because a lot of people want to invoke sexual selection for any kind of really unique feature of the body like that. Okay. And I will say that none of the ecological reconstructions I've heard have suggested that there's any way that these animals were actually using those giant teeth to feed. And it's clear that they weren't feeding on things that they would need to spear with giant teeth. They're actually feeding on microplankton. So sexual selection is a consequence of the asymmetric investment in offspring between males and females. Okay? So males need to compete to get access to the most females because males produce very little investment in each offspring, And females have a very strong investment in offspring, so females have a strong biological need to select really good mates. So you end up with this asymmetry where males are trying to get as many mates as possible in most systems, but not all systems. And females are being highly selective, and they're trying to find clues that'll tell them whether or not the males that they're mating with are going to be really high-quality males that are going to give them a better chance of success with their offspring. All right? It's all really very selfish. But in the end, if you're not selfish for your babies, what are you going to be selfish for? Okay, so, and kind of the poster boy, girl, boy, poster boy for sexual selection is this widower bird here. A widower bird called that because the females do not have the really long tails. And the really long tails mean that the males get killed more often than the females by a lot. But the ladies, they like the long tails. So they just put up with it. This is a case of runaway sexual selection. I'm going to break it down a little bit. There's actually several different kinds of sexual selection, and by thinking about the different kinds, we can actually come up with a really good idea of what's happening with the saber-toothed salmon. Okay, so the kind that we see most commonly in mammals of sexual selection is intrasexual selection, and predominantly intrasexual selection between males. My particular research area as a paleontologist has to do with these kinds of artiodactyl animals, okay? And we see over and over again in ruminants this kind of intrasexual selection where the males are competing with one another over access to the females. They're either competing for territories and have good resources that the females have to use, or they're competing directly for access to individual females or harems of females. And usually it's expressed by having these secondary sexual characteristics that are different in males and females and that have to do with that competition. It's either an actual physical competition or it's some kind of show between the two males or some combination thereof. And so we can see that like in bighorn sheep, they actually have a show that they do, and if they decide that their horns are too similar in size, then they actually go headbutting. And if we look at a lot of deer and other things, then we see similar ideas that they don't actually fight until they're convinced that they're very much the same in their level of cranial ornamentation. I wasn't sure, but this guy over here is our pronghorn antelope, which is uniquely North American mammal and the particular thing that I study the most. But I'm not here to talk about pronghorn antelope today. So there's intrasexual selection in mammals. Another kind of example here is with elephant seals. So this also illustrates one of the other things we see with a lot of intrasexual selection is this dramatic body size dimorphism between males and females. Okay, so the males are driven by their sexual selection to be really big because they have these big sumo wrestling kind of matches where they fight one another on the beach. And so the biggest guy literally is the winner. So there's strong sexual selection to be as big a male elephant seal as you possibly can. And so as a consequence, these are the males, and these are the little bitty females they're fighting over, okay? So one of the things we see with intrasexual selection is we see body size dimorphism, so two different body sizes. Another thing that we often see is very different secondary sexual characteristics, things that are expressed only in the males that we don't see in the females, like horns or like the big proboscides on these elephant seals. Excuse me. Birds don't usually have this kind of intrasexual selection. It's rare in birds, but there is a wonderful example. The great hornbill. If you want, I don't have any videos in the talk today, but if you get a chance, you should try to look up on the internet videos of hornbills fighting, because they do the same thing as the bighorn sheep, as the deer and the antelope. They actually fight. These guys fight by flying full speed into one another and hitting their heads together in the air, okay, with the giant thwack sound. And so they've evolved these big plates on their heads to protect themselves and actually change the architecture of their skulls to protect their brains. They'll fly along and just pow. It's pretty dramatic. But once again, we see body size dimorphism and we see critical differences. The females are also expressing it. And we see horns expressed in a lot of female bovids too, sheep and cattle and their relatives. And that can be a consequence of shared developmental programming. So sometimes females just have to have these things if the males are going to have them because they're developmental program. Not always. For example, most deer and their relatives, only the males have the antlers. Okay? But like I said, most birds actually don't have this kind of intrasexual selection. So intrasexual selection has dramatic body size dimorphism. Usually it has some kinds of secondary sexual characteristics that are really predominant, obvious. And this is all a consequence of males fighting with each other to get access to females. Now, the other kind of sexual selection is intersexual selection. And this is where the ladies get to control what's going on. And most of the examples of this actually come from birds, because bird biology is different enough. This is something that birds typically do. And like I said with the widower bird, what's going on here is the female birds have keyed on long tails at some point in the past, because birds with long tails that are intact are healthy, okay? But at some point, this got connected in their brains through development and evolution to the longer the tail is, the healthier the male is. And the males are strongly driven as a consequence by that same selection, so that males that have longer tails by accident get to have more babies. So there are more babies of males with long tails. And so over time, you get to the point where the tail is as long as it can be before natural selection kicks in and the males start to die off too fast to keep the species going. I've actually done experiments where they take males and they'll take the really dominant male that's getting all the mating opportunities, and they'll cut his tail off, and then they'll tape it to some other male. And then that poor bird doesn't get any more mating opportunities because he has a short tail. And the other bird that now has this super huge long tail gets all the ladies. So in the end, it's really clearly shown by experimental evidence that the female birds are keying entirely on the tails. That's the real key. But also, once the tail gets to be too long, the bird can't fly very well, and it gets eaten by its predators. Okay. There are actually a lot of different kinds of birds that have figured out how to do this thing without modifying their plumage or their bodies at all, which is maybe smarter. Although we can't really say that natural selection is smart or stupid. It just is. It does things. But in this case, a lot of for example, the bowerbirds key on this blue color. They have blue plumage. The females are already interested in blue because the males are blue. And so what they do is they find all the blue things they can in their environment, and they make these little bowers. It's a special kind of like ceremonial wooing space. And it'll put as much blue stuff as it can in there, and then it'll get the ladies to come by, and it's like, look at all the blue things I've found. And it turns out that the better bower that you have with more blue stuff, the more mating opportunities you get. And so one of the things that's happened is they've co-opted human stuff, because we make way more shiny blue things than you can find in nature. So these guys will actually go into trash heaps. They'll go into people's houses. They'll take things that are not actually trash that you're trying to use, and they'll take them and steal them away and put them in their bowers. And you can find all kinds of blue stuff in the bowers. Okay? So that's an example of intersexual selection, but it's a behaviorally modified one. And as a consequence, these guys will actually do things that are dangerous to themselves so that they can secure shiny blue stuff. All right? So it's actually interfering with their fitness as individuals because they're driven by this need to impress the ladies. This is another example everybody's familiar with of the peacocks. Okay? There's nothing at all practical about the peacock's plumage. And see, that's what the peahen looks like. A lot of people don't even know what peahens look like. They don't usually see them there. And they're not calling attention to themselves. But they don't have this huge fancy tail. That's all there to try to get her attention. And and so as a consequence, this guy has some pretty major problems trying to navigate his environment and deal with predators. But it's all worth it if he can get one look. Okay, so other cool things besides birds that have this kind of wonderful intersexual selection, there are these crazy bugs called stalk-eyed flies, where at some point, deep in the midst of evolutionary history, the female stalk-eyed flies realized that males that had their eyes wide apart on their heads were better able to see things in the environment and use their resources. And as a consequence, sexual selection has run away, and now, for no actual practical reason, they have their eyes as far apart as they can manage without actually damaging themselves in the environment. And there's actually quite a diversity of these things. And experiments have shown that there isn't actually any kind of environmental advantage to having the stalk eyes. The stalk eyes are entirely there because they look good, okay? And experiments have also shown that the further apart your eyes are, the more mating opportunities you get. So the stalk-eyed flies are more victims of runaway sexual selection. Okay, so in the end, what do we see? When we have intrasexual selection, we have dimorphism in body size, we have secondary sexual characteristics that are expressed around the mating season that are usually only expressed in the males. When we got intersexual selection, we also see some dimorphism. Often the intersexual selection only shows the selected upon character in the males, but it's not something that's practical for fighting. It's usually some display item, okay? And so when we think about salmon, what do we see? Well, here's a sockeye salmon, closest living relative to our extinct Mr. Fish. And when it's in the ocean, it's this nice hidden blue and white color, okay? Blends in. When you're underneath it looking up, you see white everywhere. That's what the underside of the water looks like when you're looking up. If you're above it and you're looking down, you see dark blue and black because that's what everything looks like when you're looking down. It's a nice cryptic. And a good mouth that's useful for feeding on the things it feeds on. When they spawn, they change dramatically. It's like Dr. Fish and Mr. Hyde. Okay, and so you get this orange coloration, you get the big hump here that's related to different musculature and things, and you get this great big hook. And the hook is there so that the males can fight with one another over access to the females when they're spawning. All right? So you actually get this dramatic change in the architecture of the skull right at spawning time. And so probably what's happening with Mr. Fish and all of his relatives is the same thing as we're seeing with sockeye. What we're seeing is some kind of intrasexual selection, okay? And we're getting all of them during their spawning time. So all the fossils are being preserved when they're adult animals that have come back in from the ocean. They're getting buried in the lakes and streams inland. So probably what we're seeing is this saber-tooth is actually a modification along like the hook on the sockeye. It's just a different evolutionary answer to the same question. Okay, so natural selection and sexual selection work on whatever's available through mutation. So if a mutation lets you do this, that's what'll happen. If the mutation lets you do this instead, that's what'll happen. Okay, so in the end, probably what we're seeing here is a combination of natural selection related to body size, optimizing reproductive ability, and also resource use in the ocean. And then sexual selection giving us these teeth that they're using for fighting for access for females once they're in the spawning situation. Okay. And we started a little bit late. So I have a whole bunch of slides talking about other saber-toothed animals. And so I want to know if I should go through that really fast. Probably take me about 5 or 10 minutes really quickly. I'm going to talk about other saber-toothed animals or whether it'd be more appropriate to stop at this point. Should we keep going? Okay, we'll keep going. We'll do it fast. We won't take too long. Okay, so this is great. This was actually an exhibit that Rachel put together at the Denver Museum. I'd really love it if we could get the same kind of thing for our museum in Eugene. But yeah, here to life size, saber-toothed cat, Smilodon, and a saber-toothed salmon, Smilodonichthys. And he's given it this wonderful tiger stripey kind of thing. I don't think that's really real, but you know, we don't know what color they were, and we'll never know that, so it might as well. But this gives you an idea of just the relative proportions of these two animals. Now, they didn't live at the same time. The critical thing is that our Smilodon fatalis is a Pleistocene animal, so it's living in the time period after Mr. Fish has gone extinct. All right? Now, it doesn't mean that there weren't saber-toothed cat-type things around at the time, just not Smilodon. And Smilodon fatalis is the one that we find at Rancho La Brea. This is a picture of a specimen we have on display at the University of Oregon Museum. It's a really nice specimen. It comes from Rancho La Brea in California from the famous tar pits, all right, which is why it's got this black color to it. Now, one of the things that I have to say, and this is part of every lecture I give about Smilodon, a lot of people want to call it the saber-toothed tiger. Please don't do that. Okay, it's not a tiger. Saber-toothed tiger would imply that it's related to actual tigers, which is not. Okay, so if you think about it, Modern tigers and modern lions are very closely related. They're sister species, and they have a very close common ancestor through time. Lions and tigers, very similar. They just have different social settings, and one of them's orange and one of them's not. But saber-toothed cat, a Smilodon, actually comes from an extinct subfamily of cats. It has a very distant common ancestor with all of the modern living cats. So it's not a tiger at all. It's actually a cat, true, but not a tiger. So please, Smilodon, the saber-toothed cat. All right, not a tiger. Was it solitary or was it social? This is always a big question. Did they hunt in packs? Did they hunt by themselves? I think one would be enough to kill me. But it's an important question scientifically because we have to know the answer if we're going to get a really good reconstruction of that late Pleistocene ecosystem, understand how all the organisms were working together. Was it operating like a tiger or like a jaguar, like any other solitary cat, or was it actually acting in a social way? And part of the reason why people even started to think that they might be social is that, the teeth of this animal are so preposterously huge that it actually goes through a phase when it's getting its adult teeth. Okay, so cats, just like humans, go through the tooth replacement cycle, that it would have basically not been able to use its saber teeth at all. And so people wondered if they would have actually been in big groups, and the group would have been able to support those juveniles as they go through the tooth replacement. But it became a bigger problem, just like I said, for trying to understand the total ecosystem at the time. And the evidence right now is starting to point to them being social animals. Okay, several studies, including one that I participated in, have shown that when you look at the proportions that are preserved in the tar pits, they're actually preserved at a rate that you would expect to see a social animal. So the tar pits are interesting. They're a trap. One herbivore, like one mammoth, would get trapped in the tar and be slowly dying. I'm in the tar. And then this kind of tar baby effect would kick in where it would be a trap for all the carnivores. And so the carnivores would come to this dying animal and they would get trapped in the tar as well. So you end up in the tar pit fauna, in the fossil record there, you end up with like an order of magnitude, like 10 times more carnivores than you would expect to find in normal proportions. And the reason why is because they're being attracted to these kills. And it turns out that the number of these saber-toothed cats that are being attracted to the kills is actually proportional to, if you go to Africa and you play the sounds back of dying animals, the different carnivores come to investigate. And the lions come in similar proportions to what we find for these saber-toothed cats in the Rancho La Brea. So when we look at an African ecosystem today and compare it to the number of these guys we're finding in the tar pits, yeah, it looks pretty clear that they're social animals. There's a bunch of other evidence too. Now the teeth of these guys something that they're doing. There's no dimorphism. The males and the females both have the same huge teeth. Okay? When we look at the juveniles, like I said, they do go through a phase where they don't have great teeth, but they actually have this interesting system where they'll have these baby saber teeth that the babies have that are still proportionately huge. And they don't fall out until the adult saber teeth have basically come all the way out. So they'll keep both of their canines in their mouths until right at the point where the adult teeth are all the way out. And so they'll end up with never having a period where they really don't have the saber teeth. Like I said, this question was from early on. We've found a lot more specimens since then. So they actually don't ever really lose their teeth. And then we see multiple convergences, and that's the real critical thing. The saber-toothed cat niche is something that seems to be an attractor in evolutionary space. We see things getting into it over and over and over again. And that suggests strongly that it's not the caprice of females looking for these teeth or males fighting with each other over the teeth. It's actually something that has to do with their feeding ecology. Okay. Other saber-toothed cats we find in the Oregon fossil record. These come from John Day. This one right here is an Nemravis that comes from Thomas Condon's collection. Thomas Condon was, of course, the pioneer geologist who founded the sciences at the University of Oregon back in 1870, 1806 when the university was founded. So this skull right here has little dirk teeth. They're not quite big enough to be saber teeth, but they're that same kind of morphology. And then this U. smilis here that also comes from the John Day, this is in the collections of the National Monument, also has these big saber teeth. And if you look, I was trying to remember if this is the specimen, and it is. This one actually has the adult tooth all the way in on the right and then partially grown in on the left. The critical thing to understand is that these are nimravids. They're not cats, okay? So this is a group of carnivores that are as closely related to cats as something like dogs are, okay? They're not very closely related to cats. They have independently evolved into this cat morphology, this cat ecological niche. And what we're seeing is that after they evolved to be like big cats, then over and over again in their group, they evolved this saber-toothed cat morphology. Okay, so this is actually a completely separate evolutionary experiment that ends up in the same place. That's suggestive that there's something very strongly important, unifying, that's not just sexual selection, it's something about their ecology that's pulling them into this place. A lot like the way that sharks and dolphins all end up looking like attack submarines because they're really controlled by the water. They have to be that shape if they're going to be efficient at swimming fast. Okay, and it's not even there. That doesn't end there. This thing right here is called Thylacosmilus, which basically means marsupial smilot. That's marsupial saber-toothed thing. Okay, so this isn't even a cat. This isn't even a placental mammal. This is a marsupial. This animal would have had a pouch, and the babies would have grown in a pouch like koalas and kangaroos when it was alive. But it's got this giant saber-tooth thing going on. It's crazy because marsupials don't have the same set of teeth that we do. They're very slightly different. What's actually happening is that this is a great big incisor. Okay, so this is growing out of the incisors in the front of its mouth, but it's still becoming this huge saber. So it's apparent that even in marsupials, they're getting the same kind of evolutionary pressure to become these big saber-toothed cat-like animals. So saber-toothed cat form has evolved a bunch of times through the history of life. That suggests that this form is actually something that's not sexually selected, that's much more likely related to some kind of extinct but interesting ecology, some kind of a killing bite that we don't have for any animals that are living today. And so as a consequence, there are a lot of people doing research trying to understand just what are all these animals doing with these huge fangs. And we're not exactly sure. There's a bunch of really good hypotheses, but nobody's been able to narrow it down to my satisfaction yet. So that is very different from what I think is happening with this saber-tooth, okay? That saber-tooth is not the same kind of killing by saber-tooth. This is something that's related to sexual selection and competition between males. So in the end, what we see with the giant Smilodonichthys, with the Oncorhynchus Smilodonichthys rastrosus, the saber-toothed salmon, is its giant size is probably a consequence of evolutionary pressure from its ecology and its reproductive biology, and that the giant tooth that we find is probably related to sexual selection, mate competition during the spawning phase. Okay? So I hope this hasn't been too dry, and I hope it's been informative, and I hope that you have a little bit more curiosity and interest in the wonderful, deep evolutionary history of Oregon, and I hope that you have a little bit more of an appreciation for how, by studying how organisms have used the earth in the past, and how evolution has shaped them through different climate changes, environmental changes in the past, we can have a better clue about how organisms are going to be shaped by our climate changes in the future, okay? A lot of people have helped me as I've done the different research I talked about here today, and you guys have helped me a lot just by being here and being a good audience. So thank you very much for your time, and if it's okay, we'll do some questions now.
So first and foremost, if anybody has any questions, go ahead and raise your hand and I will try and pass this to you. One of the things, if you happen to be here for the SWOCC Geology lecture, thank you for coming. If you happen to be here for the OIMB lecture as well, thank you for being here.
Are there earlier salmon fossils that do not have the saber teeth?
Are there earlier salmon fossils that do not have the saber teeth? Yes, as far as I understand it, we do have other salmon that are more like our modern salmon. So what we see here is that this guy is kind of an interesting evolutionary experiment on its own, and that there's other guys in the fossil record that look like they're behaving more like modern salmon. through the whole time period.
If it's hypothesized that the saber-toothed salmon had the sabers because of sexual selection, does that mean the salmons that were found in the marine locations do not have the saber teeth?
That's a really good question. So the question is, do the salmon that we find in the marine sections not have the saber teeth? Unfortunately, and I should probably have made this clearer, so thanks for bringing it up, the only place where we really get complete preservation is at that one gateway quarry. Everywhere else where we find these, what we're mostly finding are disarticulated bones from the skull, and mostly we're finding the great big vertebrae, okay? So before the University of Oregon crews found this specimen, found that quarry with these really nice articulated things, people knew that there was this giant fish in the fossil record. But it was just kind of this thing like the Bigfoot. Okay, there's a giant fish out there somewhere because they'd found these vertebrae. They could tell they were salmon vertebrae, and they could tell they were way bigger than anything ought to be. But it wasn't something that was good enough preserved, had enough information that you could name a species from it. And so most of the other sites, that's all we're finding. And as a consequence, we can't be sure whether they're having these big saber teeth or not. But that actually would be the major test of that hypothesis, would be looking for the maxillae where that tooth is located and trying to find specimens of that in those marine sections and seeing if it's present. If it is, then that rejects this hypothesis that that tooth is only grown during the mating season, and we have to reconsider, okay? So that was a really good question.
Well, we'll catch that one in a second. I'd like to thank Dr. Davis for taking time, just getting back from the East Coast and coming over today and leaving the family behind, and also suffering through the cold that you've been dragging along. We'll take one more question, then I'm sure that he'd be willing to answer some questions following that.
When your team had Mr. Fish scanned, were you concerned about possible heat damage from that scan, being it's so fragile?
That's a good question. So when I talked to the scanning researchers about it, they didn't suggest that it was going to become so hot that it would be a concern for the bones. I was actually surprised at how much warmer it got. But yeah, actually that would be an important concern to think about in the future, knowing now that it does get that warm when we scan it that way. So it wasn't something that I'd anticipated, but certainly fragile bones can be damaged by dramatic changes in the temperature, especially rapid changes. So it's something that we'll definitely have to consider the next time we go to do a scan there. Thank you.
Again, thank you all for coming out this evening. I appreciate it. If you do have a place to hang posters, there's some out there. And also, if you didn't have an opportunity to sign in on your way in, if you could do that on the way out. Definitely, if you're in my class, you want to do that.
Thank you again. I really appreciate this opportunity.